Hello, this is the second video I'm going to do on enzymes, and in this video I'm going to look at the factors which affect the rate at which enzymes are able to catalyse reactions. So in the notes, this is on page 19, 20, 21, 22, 23. Yeah, so page 19 to page 23 in the notes. Okay, so we're going to look at some various factors, try to describe and explain how those factors affect the rate. Okay, so what factors do affect the rate then? So substrate concentration, enzyme concentration, temperature and pH are the four factors that you need to be familiar with, as well as, and I'll do this in a subsequent video, inhibitors. I'm just going to concentrate on this four during this video though. This is a fermenter. So fermenters used in industry to monitor and maintain the optimum conditions for particular reactions involving enzymes. Okie doke. So to start with, if we were to describe and explain this graph, this is a pretty common type of question you might be expected to do in the exam. You may be given a graph with two axes. On this axis, we have, well, on the x-axis, the enzyme concentration. So as we move along the x-axis, we see an increase in that enzyme concentration. On the y-axis, we have the rate of the reaction. Okay, I don't know why that's. So that should say rate of catalyzed reaction. Here it's been given this value of v for velocity. So velocity just means the rate. So we can see here, if we were to describe the to describe this trend, that as the enzyme concentration increases, so does the rate of the reaction. So we have a directly proportional and positive correlation between enzyme concentration and the rate of the reaction. That would be our description. Okay. To explain it, we need to think about what's actually taking place. So, at a low concentration of enzymes, there will be few active sites. Active sites available. So, the limiting number of active sites available will limit the rate at which this enzyme catalyzed reaction can take place. As the enzyme concentration increases, more active sites become available, and therefore the rate of the enzyme catalyzed reaction can increase. We continue, more enzymes become available, more active sites become available, more enzyme substrate complexes can form. As more active sites become available. So that is our explanation here. That explanation assumes that the concentration of the substrate is in excess, i.e. there is abundant supply of substrate. Substrate is not a limiting factor. However, if that substrate becomes a limiting factor, if only if there is no excess in substrate, the rate of that catalyzed reaction would eventually plateau. So we can see at this point that there is no further increase in the rate of the enzyme controlled reaction. We get a plateau on our gradient here, and that is because the enzyme substrate complexes are forming at their maximum rate. Okay, so just to reiterate here, more enzyme substrate complexes can form as more active sites become available, because there are more collisions between the active sites and the substrates taking place in any given time. Right, the next graph that we're going to look at has different axes. So this time we're looking at the substrate concentration. Again, on the y-axis we have the rate of the reaction, and we can see initially that as the substrate concentration increases, so does the rate of the reaction. However, in this graph, it's possible to see, in some of the graphs you'll see on exam papers, there is an obvious trend that the rate of the reaction doesn't increase with the same gradients. So eventually we reach this plateauing point where the rate of the reaction no longer increases despite an increase in the substrate concentration. Okay, to describe and explain that then, so we're now thinking about a given concentration of enzyme. So at a given concentration of enzyme, i.e. the enzyme concentration remains the same at the start as at the end. Okay, initially, as the substrate concentration increases, there can be more collisions between the substrate and the active site, and we can see that here. So as a substrate increases, more collisions between the active site and the substrates, so more enzyme substrate complexes, struggling here, form. Okay, initially the rate of the reaction is limited by the concentration of the substrate. So whilst there are many active sites available, there are fewer substrates, so there are fewer enzyme substrate complexes formed. Substrate increases, more enzyme substrate complexes can form, the rate of reaction increases. Eventually, because we've got this given concentration of enzyme, i.e. the enzyme concentration remains the same, eventually those active sites will become saturated. Okay, so the substrates have taken up all of the active sites, and the reaction will continue at this maximum rate, Vmax, or the maximum velocity. And the reason for that is there is a saturation of active sites at this point. Okay, so the rate of the reaction remains high, but it no longer increases. The only method for increasing the rate of reaction at these higher substrate concentrations would be to increase the concentration of the substrates. Okay, so let's just reiterate that. Initially, there is a low rate of reaction as there are few substrates to form enzyme substrate complexes. As the substrate concentration increases, we get an increased rate of enzyme substrate complexes forming, and the rate increases. Eventually, the active sites all become saturated, so we reach its maximum rate of reaction, and the only way to increase that rate of reaction would be to increase the concentration of the enzyme. Let's have a look at the next one. Okay, here we also we have slightly different axes. This time we have time on the x-axis. So here we're looking at a reaction which is taking place over a given time. On the y-axis, so the dependent variable, we are measuring the mass of the product. So what we can assume then, at 0 we add the substrate to the enzyme. Okay. Over time that substrate is converted, or the substrate forms an enzyme substrate complex and is converted into the product. We're measuring the mass of that product as it changes over time. So initially, the product is formed at the fastest rate. So you can see in this initial period is the fastest rate. And if you think about that, that is because it's during this initial period when the substrate concentration is at its highest. So as the highest substrate concentration. Okay, if we think back to it, essentially we're looking at this part here. There's a really high substrate concentration, and therefore the rate of the reaction can be greatest. So in fact what this graph shows, the gradient shows that rate of reaction. As the reaction progresses, we can see that the gradient starts to decrease, starts to become shallower, and eventually flatten. The reason for this is that as we progress through time, more of the substrate is being converted into product. So substrate is converted into product. So therefore we have a lower substrate concentration and fewer enzyme substrate complexes can form. Eventually this gradient completely plateaus. So there is no further increase in the product produced, and this point where it's plateaued is because all substrates have been converted to product, i.e. the substrate concentration at this point is 0. Just to reiterate here, it's really important that we consider this idea, the rate at which those enzyme substrate complexes are forming. At the fastest rate initially, highest concentration of substrate, and therefore this is the highest rate at which those enzyme substrate complexes can form. As the substrate is converted into product, substrate concentration decreases, fewer enzyme substrate complexes can form. At the end, all of the substrate has been converted into product, so there are no substrate left, no enzyme substrate complexes can form. So, with substrate concentration, it can be shown in two graphs. So it may have the substrate concentration against the rate of the reaction, or alternatively it could be the time against either the total mass of the product, or in some cases, not very often, but they might have the mass of substrate here. And if it were the mass of the substrate, obviously this graph would have the opposite shape. It's not very well drawn there, but you get the idea. The substrate, mass of the substrate would decrease over time. Next, we're going to look at temperature. Okay. Here we can see the temperature is being increased, and we're measuring on the y-axis the rate of the reaction. Let's have a look at how we can describe and explain that. So, as the temperature increases towards 40 degrees, or towards this optimum temperature, the rate of the reaction also increases. That is our description. Above that optimum temperature, the rate of the reaction decreases. Again, description. To explain that, we need to consider these things. So, with increased temperature, there will be increased kinetic energy, i.e. the substrates and the enzymes are moving around more quickly, and therefore it's more likely that there will be collisions and more likely that enzyme substrate complexes are formed. So with increased kinetic energy, we get more enzyme substrate complexes forming, and therefore the rate of the reaction increases. Now this continues to increase up to that optimum temperature. However, beyond the optimum temperature, where the rate of the reaction is at its maximum, we can see this decrease. The reason for that is hydrogen bonds primarily, but also ionic bonds become broken by this increase in kinetic energy. That causes the loss of the tertiary structure. Now the tertiary structure is important in the enzymes because it's a tertiary structure which determines the shape of the active site. So a change to the tertiary structure would lead to a change to the shape of the active site. The active site would therefore no longer be complementary to the substrate, fewer enzyme substrate complexes can be formed, and the rate of reaction decreases. Okay. This is usually permanent if the temperature increases beyond 50 degrees in most enzymes. There are exceptions to that, but in most cases, when the enzyme or when the temperature of the environment in which that enzyme is found increases beyond 50 degrees, that would be a permanent loss of tertiary structure and a permanent change to the shape of the active site, and therefore enzyme substrate complexes cannot be formed. Okie doke. So, another graph that you may see, this is in your notes as well, and has been in one form or another in lots of different exam questions. It shows three lines. Oh, actually, let's look at the axes first. So firstly, we're looking at the progression of a reaction. Here we've got quantity of product formed, similar to that one that we looked at earlier on, so the mass. This is just an arbitrary unit, but if it were the mass, obviously it would be in grams or kilograms perhaps. And then it shows three different reactions. So the first reaction is taking place at 25 degrees Celsius. Actually, no it isn't. First reaction is taking place at 60 degrees Celsius. The second reaction at 37 degrees Celsius. And the third reaction at 25 degrees Celsius. But let's consider this 25 degree one initially. Okay, 25 degrees, there is low kinetic energy. Okay, so as there is low kinetic energy, there will be fewer enzyme substrate complexes forming in a given time. And that means that the reaction progresses at a slow rate. Okay, we can see it continues to increase at this slower rate as more enzyme substrate complexes form. Eventually, in fact, we'll mention this in a second. In line B at 37 degrees, so obviously 37 degrees human body temperature. At 37 degrees, there is more kinetic energy, and therefore the reaction proceeds at a faster rate due to more frequent collisions and the formation of or more frequent formation of enzyme substrate complexes more frequently. And this is in B at 37 degrees. Okay, now we can see at or in this line B that at this point the quantity of product formed stops increasing. The reason for that, as we said earlier on when we looked at this graph, is that at that point where it plateaus, all of the substrate has been converted into product. Or substrate converted to product. The substrate concentration at this point is 0, so there can be no more enzyme substrate complexes forming. Lastly then, at 60 degrees, I'll do this in a different colour I think. Okay, at 60 degrees, the initial rate is much greater, so the quantity of product formed increases rapidly initially in this early period. And that's because there is a very high kinetic energy. Therefore, the reaction proceeds faster due to more frequent collisions and more enzyme substrate complexes forming. However, we can see in this graph that the line plateaus below that point of B. All three reactions started with the same substrate concentration. So why does this line at A plateau below B? Well, we know 60 degrees is above the optimum temperature. Okay, we're assuming that 60 degrees is above the optimum temperature. So we can assume then that the hydrogen and the ionic bonds are broken. There has been a loss in the tertiary structure that changes the shape of the active site, so fewer or no enzyme substrate complexes can be formed. So to explain why the reaction stops where it does, here we can use this explanation. Essentially, the enzyme has become denatured. In a question about this, if there are very few marks available, it may be enough to say that the enzymes are denatured. In most cases, it's really important to include all of this detail. Name at least one of the bonds which will become broken. Say that the tertiary structure is changed, so that the active site changes shape. Shape's important, by the way. And say that there are fewer or no enzyme substrate complexes being formed. Okay. I said earlier on, what would happen if we continued this time for longer for the reaction at 25 degrees Celsius. We know at 25 degrees Celsius, at low kinetic energy, the enzyme is not going to be denatured. So in fact, if you were to continue that for a longer period of time at 25 degrees, eventually that 25 degree line would reach the same quantity of product formed. So at this stage, all substrate has been converted to product. So that is the maximum mass or quantity of the product formed. Okay. Last bit that we'll look at is the effect of pH. So with pH, similarly, I suppose, to temperature, there is an optimum pH at which enzymes perform their function. At that optimum pH is where the enzyme is in its correct shape and its active site is most complementary to the substrate. Any deviation away from the pH, so maybe becoming more alkali or becoming more acidic, the tertiary structure of the enzyme changes, which leads to changing in the shape of the active site. So the active site becomes altered, fewer enzyme substrate complexes are able to form. In a bit more detail, it's important to remember to mention at least one of these bonds. So hydrogen or ionic bonds, ideally say both. So hydrogen ionic bonds are broken as we have a deviation away from that optimum pH. That causes a change or a loss of the tertiary structure, changing the shape of the active site, and again that really important phrase, fewer enzyme substrate complexes can be formed and therefore the rate of the reaction decreases. With an increase in pH, we have exactly the same thing. So any deviation away from the optimum temperature, hydrogen ionic bonds are broken, loss of or change to the tertiary structure, change to the shape of the active site, fewer enzyme substrate complexes can form. Fairly common question that's linked to that then. Here we have two different enzymes, pepsin and salivary amylase. Here is their pH of the solution that they're found in on the x-axis and the rate of the substrate breakdown. Rate of substrate breakdown equivalent to the rate of the reaction. So we can see salivary amylase, this is a solid line, has an optimum temperature which is greater than the pepsin. So any deviation away from that away from the optimum pH for either of those enzymes leads to hydrogen bonds, ionic bonds being broken, change to the tertiary structure, change to the shape of the active site, and fewer enzyme substrate complexes being formed. Extra little explanation for this graph though. Why do they have such different curves? Well, you may know that pepsin is found in the stomach. Okay, released into the stomach as pepsinogen, converted into pepsin. Pepsin in the stomach has an optimum pH around pH 2 due to that presence of the hydrochloric acid in the stomach. Salivary amylase has a pH around neutral due to the pH in the mouth. Okay, that's everything I'm going to do on those 4 factors. So we've spoken about the substrate concentration, the enzyme concentration, the temperature, and the pH. In the next video, we'll look at the effect of inhibitors on the rate of an enzyme-controlled reaction. Okay, thanks a lot. Cheers.